In this video, we're going to be talking about bacteria. Bacteria in general, come in three different basic categories based on their shape. On the left-hand side, you see what the bacterial class of cocci, and they tend to be spherical in nature. Then in the middle is bacilli, which are rod-shaped, and on the right are spirilla, or that's because they have kind of a spiral shape when you look at them under the microscope. Now, bacteria are single-celled organisms, but they have the ability to come together and form what's called biofilm. And this biofilm is a collection of bacterial cells that cooperate together, and actually, when there's enough of them, when it's formed, they create channels inside these biofilms so they can send nutrients to the mass of these bacterial cells so they can all survive. Now, this is a common problem when, for example, you use plastic tubings in hospitals and for medical equipment. If bacteria gets in there, sometimes they can form biofilms right in the plastic, and the biofilms are extremely difficult to get rid of. So formation of bacterial biofilms is a huge problem, but it's a powerful way in which bacteria come together and they work together to survive. So you've probably remember that we've seen this before. This is just a general representation of bacterial cell structure. So let's take a look at some of the things that are here. So bacterial cells they have a chromosome. It's usually one piece of chromosome in a large circular form. Also, they have ribosomes. Like all cells, bacterial cells have a cell membrane, which is a plasma membrane, but they also have an outer layer of cell wall, which makes them so rigid. Now, in here, you see an extra layer outside of these two structures, and usually this is called capsule, and usually you see these capsules in pathogenic bacteria, and that helps bacteria become really robust. Then we have a structure called flagellum, which allows bacteria to move around. This is similar to that tail of a sperm that you see in higher eukaryotic cells. Then these structures called pili, these are used by bacteria to transfer genetic material between one another through a process called conjugation. Now, bacterial cell walls come in two different varieties, and they are basically under the microscope, they are recognized by the fact of whether or not they can stain with a particular stain called gram. So you have gram-positive bacteria. These are bacteria which can be stained with the stain, and gram-negative bacteria where you can't stain it. And there are important differences between the outer layer of these cells. As you can see, gram-negative bacteria only has one plasma membrane, and then the outside of this plasma membrane there is a multiple layers of what's called peptidoglycan cell wall. So peptidoglycan is a combination of carbohydrates and short peptides. So you have a row of carbohydrates here and here and here, and they are connected through short peptides, as you will see shortly. Gram-negative bacteria have two plasma membranes, the inner membrane and an outer membrane, and they are separated by a thin cell wall of one row of peptidoglycan. So now let's take a look at this peptidoglycan multi-layer wall in gram-positive bacteria. So here's a blow up of what would the outside of this bacteria would look like. Here's a model of the plasma membrane, the phospholipid bilayer, and then you have rows upon rows of peptidoglycan layers. So this is one layer, that's another layer. So these layers are stacked on top of one another. Now, these carbohydrates, which are of two variety, an acetylglucosamine and an acetylmuramic acid, they are connected to one another end over end, and then the layer is formed by connecting these chains of carbohydrates through little short peptides in here, which are connected together through another piece of short peptide. So, and you can see in here the legend is describing it. It's two tetrapeptide chains. So tetra means four. So there are 4 amino acids attached together in each chain, and they are cross-linked by another short peptide. So these peptide chains are really important for holding these layers of peptidoglycan cell wall together. It's essential for survival of the bacteria. Which brings us to the discussion of an antibiotic, which is penicillin. Penicillin basically prevents growth of bacteria. It kills them by disrupting this peptidoglycan cell wall. So let's see how that happens. So normally you have these rows, these peptidoglycans, as you see here. Okay. So these peptidoglycans, these ones, these carbohydrates that are attached together end over end, they need to be held together, and they're held together through these peptides. But in here, you can see that these peptides aren't connected together yet. And that connection is really important to hold these chains together. There is a protein in bacterial cells called penicillin binding protein. The reason they call it is because it binds penicillin. But this protein has a normal function in bacterial cells, and its function is to form these covalent bridges between these peptides. So you can see that it's coming in, and it is catalyzing the formation of this short peptide, short bond, this covalent bond between these peptides. So this is a short row of glycine amino acids. There's 5 of them, which is sitting in here. Which are sitting in here. Now you need an enzyme to connect these peptides together through this short little peptide, and that enzyme is the penicillin binding protein. So the penicillin binding protein comes in here and connects these peptides together, and it does that one over another. So it forms a covalent bond here, then it goes to the next one, such that in the end you're going to get these connected together through these peptide connections. Okay, so how does penicillin work? Penicillin is a competitive inhibitor for the substrate of these enzyme, which is these proteins that are sitting here. So what penicillin does, it comes in, it binds to the penicillin binding protein and stops it from forming these covalent bonds between the peptides. And therefore, it destabilizes the cell wall of bacterial cells and kills them. So one fun fact, lysozyme is an enzyme which is found in tears. So when we cry, we have these lysozyme that is included in them. Now, lysozyme is able to break the bonds in the peptidoglycan cell wall. So it is actually this enzyme that is in our tears, it's an antibiotic by itself, and it's considered as part of our immune system. So how do bacteria reproduce? Bacteria reproduce by binary fission. That's basically what the mechanism is called. So you start with one bacteria cells, then the bacteria makes a copy of its genetic material. These little guys are plasmids, which we learned about in the biotechnology lectures. And they are these pieces of genetic material. They are copied, and then they move towards the opposite ends of a cell, and this cell basically gets longer and longer, and it pinches, and then it separates. So that's called the binary fission. Now, bacterial gene transfer can happen in multiple ways. One of them is transformation. Bacterial cells can, on a random basis, if the conditions are met, can just take up DNA from genetic material from their environment and incorporate it into their genome. Or if it's a plasmid if it can replicate independently on its own, it can just be independent out, which is not incorporated into the chromosome. And we saw this when we were talking about cloning. We use transformation to get recombinant plasmids into cells. Another way is transduction, which is through the use of bacteriophages. Bacteriophages are viruses for bacteria. So bacteriophages carry genetic material and they infect bacterial cells, and sometimes these genetic material of the bacteriophage gets incorporated into the genome of the bacterial cell. And the last mechanism is through conjugation, which I talked about through those pili. These genetic material can travel between one bacterium to another through the channel that is formed by those structures. So we have good bacteria and we have bad bacteria. So let's take a look at what bad bacteria is. And the general feeling about bacteria in human society is that bacteria are all bad. Actually, not all of them are bad. You probably already know bad bacteria cause disease, and we're just going to talk about a few of them. There are many. One of them is Yersinia pestis, which causes what's called the Black Death. And this Black Death has a very, very interesting historical story to it, which you can watch it by clicking on this video. I'm going to share the link to this video on Canvas right after posting this lecture video. So I encourage you to watch it and learn a little bit about this bacterium and the events that happened around the Black Death in history. Cholera is a kind of bacteria which really makes people sick and they can die. And usually it is abundant in areas where clean water is not present. And cholera, there tends to be outbreak of cholera whenever there is flood and no sanitation. And this map shows areas of the world where cholera cases exist. So in places where there is good sanitation with respect to the source of water, you really don't see cases of cholera. Foodborne diseases, bacteria can cause foodborne diseases. This is one example of a strain of E. coli. There was an outbreak of E. coli in Europe, which sickened and killed many people. And this is an electron micrograph of E. coli. And this one shows the vegetable that was grown. These sprouts, vegetable sprouts, were infected by E. coli. I'm sure you've heard about the breakout of E. coli in lettuce. Once in a while, we have that in romaine lettuce. So that's another example of bad bacteria that gets into our food system. So now let's talk about good bacteria. They're actually good bacteria play a very, very important role in our well-being. And the collection of bacterium, and not only bacteria, but there's also yeast. It could be yeast that live within our bodies and also exist on our skin. They are collectively called the microbiome, and they play a very important role in helping our digestion. They play an important role in our immune system. There is a video in here that you should watch about the importance of microbiome in our survival. I will not click it here. I will place the link to this video on Canvas, and you are required to watch it. So this is not a video. It's not something you can skip. Another important role for bacteria is helping us make processed food. And I don't mean by factory processed, chemical processed, but traditionally bacteria have been used throughout the human civilization to help us make different kinds of food, like cheese, salami, yogurt, and apparently fish sauce. There are some bacteria actually that can metabolize oil. So these are very useful because we tend to have the habit of polluting our environment by digging up fossil fuels and spilling it out into the ocean. And bacteria are used for bioremediation. So whenever there is an oil spill, there are some species of bacteria that are going to be brought to the regions of oil spill, and they're introduced to the region. And because they can break down the oil, they are a very useful tool to help us correct our mistakes. Another important and essential role that bacteria play in our survival is nitrogen fixation. Now, nitrogen, if you remember, is an important and abundant component of molecules that are used for life. So in amino acids, every amino acid has an amino end, if you remember. They have nitrogenous bases in our DNA. So nitrogen is one of those elements that is one of the 4 types of elements that are the major components of life. But most organisms cannot fix nitrogen from the atmosphere. So there is nitrogen in the atmosphere in the form of nitrogen gas, N2. But in order for living beings to be able to take that from the environment and use it, they have to be able to fix it. And pretty much the only type of organisms can do that are nitrogen fixing bacteria. So here in this example, the nitrogen fixing bacteria, a certain kind of nitrogen fixing bacteria live in a symbiotic relationship, and symbiotic relationship is in a form of a relationship where the relationship benefits both partners. So you have a plant in here that requires fixation of nitrogen from the environment so they can use it and make their DNA and amino acids and whatever. And then we have nitrogen fixing bacteria which can fix nitrogen from the environment, but they cannot grow in the presence of oxygen. So there are regions within the roots of these plants where there's very little oxygen. So these bacteria can have a region where they can live in an environment where they can survive, but they can fix nitrogen. So in the exchange from having a place to hide and taking advantage of the fact that this plant makes food through the process of photosynthesis, these bacteria can live in here, and plants can take advantage of the fact that these bacteria fix nitrogen. So nitrogen fixation happens. It's fixed from the atmosphere, and then there are also, in addition to these specific types of bacteria that work with the roots of legumes, there are other nitrogen fixing bacteria that can also fix nitrogen without the partnership with this root. So they turn nitrogen into ammonia. And then there are some other bacteria. They turn ammonia into nitrites and then nitrates, and then these nitrates are incorporated into the plants. The plants can use it in order to form the building blocks of their biological molecules. Now, other organisms, so for example, in this example, it's a rodent. The rodent comes in and eats the plants, and then this rodent at some point dies, and then the decomposers are going to decompose this mice and return the nitrogen that was present in this mice. They return it back to this soil. So there's lots of different types of bacteria that are involved in capturing nitrogen and giving it to other organisms such as plants. But nitrogen, some of it needs to go back into the atmosphere. So that's why we call this the nitrogen cycle. So there are bacteria that actually are called denitrifying bacteria. They take this nitrate and turn it back into nitrogen and give it back into the environment. So there is a really nice balance between the nitrogen fixing bacteria, which fix nitrogen from the environment, and the ones that give it back to the environment. So I hope this image helps you appreciate the important role that bacteria in the soil play in the survival of all of us.